8:35. Good morning, everybody. My name is Li from Electrical and Systems Engineering at Washington University in St. Louis. It is my great pleasure to welcome all of you this morning on behalf of the organizers, Dr. Chin and myself, and all of us at the School of Engineering and Applied Science to our inaugural workshop, Brain Dynamics and Neurocontrol Engineering. This workshop aims to provide a focused forum for the discussion of research synergy between experts from dynamics, control, and neuroscience communities. And let me explain the background of this workshop briefly. This workshop builds on storied history of system science and mathematics at Washington University, which is a unique program specializing in applied mathematics, systems, dynamics and control, and operations research, and promoting transdisciplinary research. And building on our collaborative culture and institutional strength, we have continued to push the envelope of system science and engineering in diverse application domains, including biology, medicine, biomedical sciences, and biomedical engineering. And this workshop embodies our forward-looking approach to system science as a discipline not limited to the modeling, construction, and design of engineered systems, but a framework upon which to understand the engineering within the nature and human societies. And neuroscience offers great new and interesting scientific challenges due to the immense complexity of the system at hand, the dynamics of which span multiple spatial and temporal scales. Understanding how these dynamics mediate brain function is a pivotal neuroscience question that is seemingly well aligned with the approaches innate to systems and control engineering. Therefore, this workshop is intended to open a dialogue between experts in systems and basic science in order to facilitate a discussion of how recent developments in systems and control theory may stimulate new approaches for controlling neural circuits, and furthermore, how they may provide new frameworks for basic analysis and interpretation of brain dynamics, function, and networks. So in more general terms, by facilitating the discussion early in the inception of this interdisciplinary area, the workshop will aid in ensuring long-term, sustained research impact. So here, I sincerely hope that the discussion in this workshop and the information shared can be applied effectively to your future research activities. In closing, so may I acknowledge with gratitude the support we have received from the National Science Foundation, and the program officer, Dr. Jordan Berger, is there, and the fund from McDonald Center of Systems Neuroscience, Burroughs Wellcome Fund, and the School of Engineering and Applied Science at Washington University. I would also like to thank the great support from our Dean, Aaron Bobbitt, who is unfortunately not here today. With these few remarks, I thank you for your kind attention and declare the workshop officially opened. And may you have a productive and successful workshop. Okay. So now I'm going to hand this over to my colleague Shinnon Chin. Shinnon is going to make a few more comments, and he's going to go over the logistics of the workshop.
All right, thanks, Jershin. So let me echo Jershin's comments, welcoming you all here to WashU. I think it's going to be a beautiful couple of days of weather, so for those of you from out of town, please enjoy your time on campus. So, just to kind of build on what Jershin was saying, I think, the past couple of years have witnessed a lot of growth at the intersection of engineering, neuroscience, and brain medicine, and I do think that there's tremendous potential for knowledge creation. Combining these disciplines, and that's evidenced, I think, by the major initiatives that we've we've seen at the federal level, the Brain Initiative and so forth. And the National Academy of Engineering has as one of its 14 grand challenges for the next century the reverse engineering of the brain. So it's my belief, and I think one that's shared by many of the researchers in this room, that system science and control theory can actually play an important role in this overall effort towards both new scientific understanding and hopefully as well potentially practical solutions for clinical challenges as well. And I think that in the next day and a half, we hope that we can have some critical discussion to engage the question on how to best capitalize on this potential. So I'll just talk a little bit about the logistics. So you all have the copy of the agenda in your programs. It's pretty self-explanatory. So we've organized the presentations into 3 sessions that span topics of control, neural circuits, and then using dynamical systems and control principles to understand the brain essentially. And so it's highly appropriate, I think, that as we start our program this morning, we open with a researcher who has literally written the book on the emerging intersection between control theory and neuroscience. So it's my pleasure to introduce Professor Steve Schiff, and you all have his bio in your packet, so I won't read from it too much, except to say that I've always been, in my interactions with Steve, impressed by the breadth of his interests, and in addition to being a pediatric neurosurgeon, has pursued a robust research program in dynamics and understanding dynamical properties of the brain in the context of disease. Just a few weeks ago, we were having a conversation. He was explaining to me how he had been debugging some MATLAB code. I think that sort of combination of disciplines is exactly the type of sort of synergy that we're trying to explore in this workshop today. In addition to his research in understanding the dynamics of the brain, he's recently received the very prestigious NIH Director's Pioneer Award for his work on global health and understanding neurological disease in Africa. So I'm really delighted that he could be with us here this morning. Steve, please.
Thanks very much.
Thanks a lot. Well, thanks very much for the invitation, and I'll try to keep this a bit informal, and I'm not going to try to use a whole hour to talk at you. And I didn't know what title to give the workshop, so the complex uncertainty is mostly my confusion about doing anything that seems to involve the brain. But I'll tell you a little bit of a story. And I've been interested in diseases like epilepsy for a long time. And how can you do better at treating what are dynamical diseases of the body, short of what we do in intractable cases, which is to carefully cut out enough brain so that the phenomenon stops or gets better. And I've always been convinced there's a better way of approaching such illnesses. And the first issue is how we use models to both look at phenomena in biology, and what we haven't done over time is what the engineering community has done in the last 50 years or so of modern control engineering, which is not just to simulate things, but to actually use the models in real time to look at and observe or control systems. Rudolf Kalman put out his classic paper in 1960, where he envisioned a dynamical system, which, let's see if I can get this to work, and a dynamical system that was iterative with a formal observer placed between you and the state of that system. You don't actually touch the underlying variables. And then uncertainty or noise, but also model uncertainty in terms of both the outputs and the actual models that you use. Kalman, in this one paper, actually defined observability and controllability. It kind of summed up most of the concepts that I'll touch on today. His original version of this took uncertainty that had a bell-shaped curve and pushed it through a linear system, and you end up with another Gaussian curve. And the problem with this, and the problem with most brain dynamics, is there's very little of it which will either fit a linear system or allow you to linearize around some particular point. And so you're stuck taking lots of estimations of where you might be in the state of a system and pushing it through and getting some complex things out, because the functions you assume in neuroscience are all floridly nonlinear. Move ahead to the 1990s, and I think one of the really key developments wasn't just the development of Monte Carlo techniques that would allow you to buy an incredibly large computer and to simulate lots and lots of initial conditions, but the sparse techniques. And there's a variety of ways of doing this, and I don't think it really matters how one approaches a complex system. But in this work from Juliet and Ullman, and if you're a grad student, what you don't want to do is anything this unique and delay actually being able to publish your thesis for as many years as they did. But one can take the dynamics of a system which aren't linear and push through an optimized set of possible conditions. These sparse ensemble filters really enabled us to do a lot of the very cool things that you see in modern robotics. And in this worldview, one can also assume that what you measure things through is also nonlinear. So it's a very general approach to use when you don't want to linearize a system and you want to interface with it. Then you make a measurement, and you do what Kalman told you about, to basically use Bayes' rule to do a weighted average between your measurement and what you think your uncertainty is, and then recursively do it over and over again. So these are fascinating tools. What I love are these kinds of images, though, of what you're really doing. You measure a system, and for a brief period of time, you have the closest knowledge you have of what that system does. And then every increment in time as you move forward increases your uncertainty until you make new measurements. This is true whether you're looking at brains or the state of the atmosphere and you're doing weather prediction, or you're a robot and you're trying to figure out where you are in this building and you feel lost. You never quite know where you are, but you have an underlying model, and if you're a robot, you have a map of the building, hopefully. A couple of other concepts that I'll throw out today. A little while later, a couple of years after Kalman, Leuenberger published a well-known paper on observers. Observer systems are models of a system that allow you to do a number of things, but one of them is to reconstruct variables that you can't measure or perhaps can't access. So if you're working with the brain, you've got no shortage of things you can't access. Often we go and use measurements of voltage. Everyone loves voltage. There's a lot of other stuff going on in brains in addition to voltage, and I'll touch on some of that today. And truth be told, Kalman had laid down most of the principles of observers before this paper I'm citing. The other piece is a work by Conan and Ashby, which is, if you really want to control something, you probably need a model that represents the dynamics to do it optimally. If there's a paper I talk about today that probably needs a redo, I think it's probably that one. And since the 60s or early 50s, in parallel with developing what in engineering is modern control theory, we have taken a variety of models of individual neurons and sheets of neurons in cortex or deep structures in the brain, and made more and more efficient models. Now, these models are largely fundamental in that they take what we know about neuroscience and embody into the models and equations that fundamental knowledge. I'm not going to dwell on black box models. You can do that, and perhaps fortunately or unfortunately, you can do that with increasing power these days as machine learning gets to take over our conversations, I think. But reconstructing what you can't measure is ubiquitous. If you look at Eugenia Kalnay's wonderful book on data assimilation, I've always been struck just by how sparse our measurements are that go into our global weather prediction, short-term and medium-term prediction. The best is satellite temperature, but even that isn't complete. And we reconstruct every 6 hours at the various global agencies in the US, in the United Kingdom, and others. The picture of dynamics that fills in these gaps. If you look at what we typically do, you have wonderful pediatric and adult epilepsy surgery at Washington University for many, many decades. We think as surgeons this is a lot of coverage. There's a lot more brain that doesn't get touched by electrodes when we go and try to sense the dynamics of people with intractable epilepsy and reconstruct what might need to be cut out or stimulated in order to reduce symptoms. As was mentioned, I went a little overboard and actually put a book together from the graduate coursework that I'd been teaching. You can take what's available now in computational neuroscience and fuse it with the whole machinery of control engineering. You can do this to do the typical things, such as observing or predicting or controlling systems. I'm particularly interested in control of pathological states. The other feature to this, which isn't well discussed, is sustainability. And you can view that in a number of ways, and there's a number of ways to define what I might mean by sustainability in either control or health engineering. But these tools enable you to optimize processes. If you're going to build a device to modulate epilepsy or Parkinson's disease or any of the other conditions for which many of us are very interested right now in modulating, then if nothing else, you'd like the battery to last a long time. But you'd also like the function to be optimized. How do you make things that work the best? How do you reduce costs? A lot of our electrical stimulation strategies have reduced themselves to an extraordinary minority of the patients on the planet because we benefit here in the way we do medical care device design by constructing devices that maximize profit for the companies involved. That actually minimizes the reach around the planet for most of the people with the diseases that you'd like to affect. So part of this is also how do you get the maximum outcome benefit for the available resources you can bring to problems. All of this, I think, fits in a control engineering framework. But I have a hammer now, and so a lot of things start to look like nails to me. Can you take all of this and address brain diseases? And all right, I've been looking at what I can do electrically to annoy seizures for a long time. And somewhere about 10 years ago, the machinery of model-based control, models that will link to a data stream, absorb measurements that you have, reconstruct state. What does that mean? It means it synchronizes to the system. It means if you have the model right, it also is revealing you things about that underlying system which you can interrogate through the model, even if your recording points are partial. In a control system, what that tells you is you can control the model and the rest of the system comes along with it. If you construct an airplane autolander, you know so much about the airframe. Boeing spent a lot on this, so did Airbus. You know so much about the dynamics and the equations of motion of the airframe that the model is well synchronized to it. Even though the measurements are sparse, you can reconstruct the airflow on the rest of the airframe. You control the model, the airplane goes along for the ride. You'd love to do that in the nervous system. And I think that as time goes on, we will undoubtedly get better. The difficulty in doing this, of course, is at present, how much do we really know? So I envision this as sort of crossing lines. At present, our ability to take fundamental models often and expectedly won't be as good as empirical models based on data. You don't have to go to neuroscience grad school to do that. Get a computer science degree. But at some point, being able to bootstrap those models by actually what we do understand about the nervous system has to do a better job. That's a bit of philosophy, but I'll drag you through a few points here along the way. So in 1952, Hodgkin and Huxley published what we've used for the last 70 years or so as canonical equations describing how neurons generate spikes. And there are 4 variables here, and almost always we measure voltage. And from this master current balance equation, everything else that's in there, the conductances to ions, the actual gating variables involved, these M's and N's and H's. And if you have no idea what I'm talking about, trust me for a moment. These are curve fits. It's based on physiology, but the average curve fits from experiments that they handpicked to go into this were up to their judgment. Sometimes judgment is quite good. So we measure voltage in these things. So these are some of the first explorations that I and my students and postdocs looked at in terms of taking control engineering machinery and going back to some of the classic work in neuroscience. So what if you took just the voltage measurement, that's what we all know and love, and you add some noise to it? Could you invert this problem and generate the rest of that set of equations from the page before? So Ghanem Ullah, who's now on physics faculty at University of South Florida, and I were looking at this, and he wanders in one day, and this simply looks too good. And we both pounded on it for a while. Couldn't make this result go away. In retrospect, you all can ask me why this works. Why can I reconstruct the rest of this set of coupled equations so well if I only measure voltage? Some of this is a lack of symmetries in terms of the way the equations are put together. And that's a deeper question in observing any system if you want to do it accurately. And some of this also is look at how slowly these things over here on the right converge. So these are parameters. I'm actually doing what is known in control engineering as a dual estimation problem. I'm not just trying to reconstruct variables. I'm also reconstructing parameters. In a simple worldview, parameters are slow variables. I use a trick here, which if any of you are fans of Gil Strang's books, he wrote a book in the 1980s on applied mathematics that those of us of a certain vintage don't loan out to our students for fear it would disappear. And he talked about how to model parameters that slowly change. very insightful fellow. You could also change some of the external parameters that set the membrane potential of such systems, like potassium, something I'll come back to later in the talk. You can reconstruct an arbitrary function of potassium through this system. You get an awful lot out from measuring voltage. Now, I'm doing this in simulation. I actually have the model, and then I put that same model into my control framework. In this case, I'm using an ensemble Kalman filter, so I've cheated. But you've got to start someplace. Here's experimental data where I don't have the model. So I did some work with Xiaoying Huang in Jinyoung Wu's lab at Georgetown. Fantastic experimental laboratory. So we found that there are spiral waves in these tangential sheets of cortex that we were studying. And you can take models of cortex and do a very good job, not just of reconstructing variables and parameters, but of actually watching the error in short-term convergence get very small. What have I done here? Well, it's a whole lot better than picking a really inappropriate model, but am I actually giving you the state of the system? No. Am I giving you short-term predictability? Yes. Can I use this in a control scenario? I don't care how you give me the predictability. I only need it for a short period of time. These are issues that all of which become important if you're taking the machinery of control engineering and trying to port it over into a nervous system. We didn't know it, but at the same time, our colleagues at Maryland, this is Cornick and Brian Hunt, Ed Ott, and others, have been trying to do the same thing with Rayleigh-Bernard convection. So this is fluid between two plates. You warm the bottom one until you get unstable convection rolls appearing. You get very complex patterns out in such experiments. And they didn't take the Navier-Stokes equations. They took a simplified version. These are Bosonesque equations, just as we took a simplified model, Wilson-Cowan equations, of cortex to do the thing on top. And again, they can absorb data. They used a different version of the ensemble filter. Ed Ott has this beautiful local ensemble filter algorithm, which I encourage everybody to look at. It's a 2004 paper of Ed's. But really, we accomplished the same things. This actually was the first lab experiment looking at data assimilation in a convection experiment. And a few miles away, we're doing this on brain. So what I'm talking about is a very general approach and strategy to any spatiotemporal system. I'm more interested in brain than fluid. And Parkinson's disease, I suspect, is going to be the place where these principles will have the most near-term impact. And I say that because if you're looking at some of the symptoms of Parkinson's disease, tremor is what you ordinarily see. It doesn't disable people as much as the rigidity and the difficulty in initiating movements. But we know a lot about what happens in the static network, shown over here, in comparison with the changes in excitation and inhibition between these nuclei deep in the brain in patients with Parkinson's disease. And going back to what I said about Leuenberger, and interrupt me if either what I'm saying is incomprehensible or wrong, or you're just falling asleep and want to change course here. But the basic philosophy in Parkinson's is I've got an interacting network. Do you want me to go as a surgeon and put electrodes in multiple nuclei deep in your brain. There's a cost, even when there's no bleeding or anything, to just inserting electrodes. You get a lesion, small micro lesion effect. You can see patients change in their behavior. Let's say you're watching their tremor and movements on an operating table as electrodes get placed into position, no electricity being turned on. So if I record from a large enough sample of these sites, I will damage a brain in multiple fold ways beyond what you really want me to do. What you want me to do is to take this control engineering approach, have a good enough model, and tell me I get one shaft, I get one electrode, one pass. I can have multiple contacts. But then, if I really do understand that network, use what I'm sensing from one or more contacts to do an optimized reconstruction of its dynamics and deliver stimulation. Again, 2 contacts on that single shaft. So I don't know any other condition better set up for the people in this room from a systems engineering perspective than addressing Parkinson's symptoms in this way. How do you improve neuronal models? I wish I could say I did this from good planning and hypothesis. This is a great example. Is there someone from NSF in the room? This is a good example of where if we were to wait to generate the hypotheses rather than having people tinker around in maybe a well-prepared fashion, you'd never see people stumble into this. So I call it unification. Maybe it's my homage to Julia and Ullman. If you really want to prevent and delay publishing a paper, and you're not crazy, put the word unification in the title and start sending it off to your favorite journals. What I'm going to talk about is the different phenomena in the brain, spikes, seizures, and a phenomenon known as spreading depression. No one wants to use that word anymore. Aristides Leal from Brazil was at Harvard University in the 1940s, and he wants to investigate seizures. Remember, seizures were only electrically recorded in the 30s. So he's trying to investigate seizures in cortex, and instead of seeing seizures, he sees this wave of silence slowly propagating through his brain experiments. He calls it spreading depression, and in recent years you'll see a shift by the community to call it spreading depolarization, because people couldn't stop confusing it with cognitive depression in mental health. And now you see equal use of those terms in the literature. And then the term I think is great, brain tsunamis have been coming out. There's a website on that. You can look up brain tsunami. It's actually not a bad website. We always treated these as separate phenomena. Leal is disappointed. He wants to study seizures. He became very well known for his discovery of this phenomenon. And it's becoming increasingly important clinically. First of all, this is what generates, so far as we can tell, most of the migraine auras in people with classic migraines. So people who, if any of you have these, where you will get a slow spread, let's say, of a curtain through your visual field, or if you get scintillations, little sparkling things across your vision before you get a migraine headache, this is what's doing the propagation. So there's a lot of people with migraines in the world. We also in recent years have found that these waves of depolarization occur in the brain around a stroke, if you have a hemorrhage, not necessarily what we would call a stroke, but a hemorrhage into the fluid around the brain. If you've had a traumatic brain injury, you can get repeated periodic spreading depressions in brain, and it's really bad for the brain. people who get these do worse and have more brain damage than people who don't. Mike Rogowski, though, for years has been pointing out that not only do some of the same drugs work for both, but there are specific genetic mutations. There are 3 that I know of now. I'm sure there's 4 and 5 that I haven't yet kept up with. But these are called familial hemiplegic migraines. They're pretty rare. You may not have heard of that. But these people, same neurons, generate both seizures and migraines. So we knew that there was some way to do both, but we sort of ignored that. Interestingly, the gene defects, and I listed a few of them here, in calcium channels, in the sodium channel, in the sodium-potassium ATPase, are exactly what the following modeling would reflect. But this is all retrospective explanation on my part. Go back to Hodgkin and Huxley. They give us this model. It's probably the framework that we use in 95 or more % of our fundamental modeling of neuronal processes. There is no ion flux dynamics here. We assume that these neurons are just sitting in an infinite bath and they exchange their ions, but none of the concentrations change with time. It's a perpetual motion machine. There's no energy required to run the Hodgkin-Huxley equations. They don't generate seizures. You have to kind of hook them up into a network to get all this to happen. But I can go into the lab and get things that look like seizures from sticking single cells. They don't give you spreading depression. It's a perpetual motion machine that makes spikes. And we've been pretty happy as a field with that. So I'm going to show you a way to extend the equations based on biophysical structure and conservation laws. So there's been a real lack of conservation. And I'd love to say that this is what I plan to do. No, we were looking for a better way to model spreading depression. A student with me at the time was Yina Wei, a marvelous student, out with Maxime Bazanoff now as a postdoc. And we just kind of said, well, let's put in some more of the structure. So there's 3 lessons from this. The first one is that the extracellular space matters. What is that? Hodgkin and Huxley, when they did their papers, knew there was an extracellular space. This is actually what it looks like around a squid giant axon. It's a little tortuous thing in a glial sheath that surrounds that neuron. They stripped it off for the experiments before World War II that they came back to do those famous papers for the equations that I just showed you. But they came back in the 50s. This is from a paper by Frankenhauser and Hodgkin. Amazing paper. They left the sheath on. Single action potentials will bump the extracellular potassium by about a millimolar for a short period of time until it's pumped out. If you stimulate that axon at 10 or 15 or more cycles a second, you actually get up to a ceiling that we see physiologically where you'll raise extracellular potassium by 10 or 15 millimolar. These are extraordinary numbers, and most of us have simply ignored that. But neurons live in a very thin conductive shell. And almost everything you read, if it doesn't take that shell into account, you need to be a little suspicious whether it has to take that shell. And I don't care if you're looking at electric and magnetic field interactions, so-called activation functions, or you're looking at any of the dynamics of these neurons. If you're a student in the room, next time someone in computational neuroscience gives you a talk with naked neurons in infinite spaces, raise a hand and ask them why. It'll annoy them, if nothing else, but there's some serious reasons to raise some questions. The second is conservation. I'd love to say that we discovered how to write these conservation laws, and a little bit after we were doing it. In the cardiac literature, I mentioned one of the key authors here. In the cardiac literature, they've been doing this for years. There are multiple good papers dealing with conservations of particles and charge across cardiac myocytes that are active. They make slow action potentials, and also in control paradigms. But if you study the nervous system, there is absolutely no reason why you ever walk in to a neighboring building or a neighboring floor and talk to people who are in a different discipline. So we really have a problem with that in many areas of science. And this is a good one, where the lack of crossover and discussion between parallel literatures really kept some good ideas away from us. We have computers that run faster and faster. So this looks like a frightening bit of equations, and all I'm doing is taking the rate of change of each ion outside and inside of a cell and repeating it and writing down where the ions go. There's no reason that I can't keep track of particles. I don't want to run this in real time in this form, but I can certainly now keep an eye on how ions cross those membranes. And then oxygen. We've ignored oxygen dynamics. And one can set up the dynamics of oxygen based on the activity of what eats it up. Most energetic, the number one process in active neurons is the pumps to push particles back where they belong. And of course, oxygen diffuses and comes away from a region of brain, from the vasculature, or if it's an in vitro experiment, from a bath. We know the biophysics of this pump. If you have no oxygen, it doesn't work. Then it, like so many other processes, if you increase oxygen, you see this sigmoid-like response function. The last piece, size matters. I've got neurons in a tiny little conductive shell. I don't have to spit many ions out to get a large change in ion concentration, and the gradients of those concentrations drive the dynamics. So I can go back to freshman chemistry and add up the ions outside and inside, calculate osmotic pressure, do the simplest first-order kinetics. I stole this from a brilliant series of papers by Kager, Wadman, and Samjian, and let the neurons expand and contract. You're active. Not only do you dump some ions outside the cell, but then you swell the cell slightly, and you really amplify these small changes. So what does that look like? You take a naked neuron and you enwrap it in a thin space and surround it by these glial support cells that Frankenhauser and Hodgkin left in, and you put on some pumps and you go and let it run. What we end up noticing is the following. If you have a parameter space of oxygen and potassium, and way up in the corner here, normal oxygen, low potassium, that's the space that Hodgkin and Huxley lives in. It does all the normal dynamics of Hodgkin and Huxley. And if you allow potassium, keeping oxygen normal, to rise, it's very easy. We've been doing this ever since Steve Trainalis and Ray Dingledine did this in the late 1980s. You get periodic seizures. They look like electrical seizures in a piece of brain. And if you move along to the right and you increase potassium instead of to 8 millimolar up to the mid-20s, then you generate what David Andrew and his colleagues have been studying for a number of years in Canada. And if you then go up, fall off this graph, and you go out to around 40 millimolar, Brian McVicker has also been looking at periodic spreading depression. And if you then go and drop oxygen here, you end up replicating this mixture state of seizure and spreading depression. This lovely stuff from Gaberchek and George Samjian characterizing these states. Zant and Michel Levan-Putin published a very nice paper or two on what they call the wave of death. I call it WOD for short here. But if you take enough oxygen away, then the neurons will spill the gradients and eventually die. You get an awful lot of physiology out of what has been heavily validated experimentally by throwing in a little bit of the anatomic structure and some conservation laws. These models are simple enough, though. You can put them through. I simplify this just a little bit. If you look at the continuation methods to study the stability of bifurcations in coupled equations, most of that's based on a program algorithm set called Auto. This is using Bard Ehrmantraut's XPP, all of which enable you to take a model, slowly vary, for instance, potassium, and to look at the bifurcation structure. I think this is very important. First of all, the underlying mathematics give you this pattern of stability with spikes, seizures, slow oscillations with spreading depression, and predict an eventual fixed point where nothing's happening. You can see all of that experimentally, but this gives you the potential to take this 12-variable system with some nasty-looking housekeeping equations and reduce it to simple normal forms. I think that's important. We can get back and talk about this a bit later in a discussion, but I don't think any of you want to take something even this complex and try to use it in a control system scenario. But these simple polynomial equations that underlying the normal forms of these bifurcations are just dirt simple to use if we can work out how to interface them properly with biological systems. Another thing we did is look at volume. Little tiny changes in volume will actually dramatically alter the dynamics of this system. So there's another paper here referenced showing that one can use volume as a bifurcation order parameter here. It also helps explain what we have seen for years in terms of a physiological ceiling, whether you're Frankenhauser and Hodgkin or you're us looking at intact brains. Unless you've got spreading depression, you never see certain ions go beyond these level ceilings. 15 or so millimolar is the most you'll ever see in a brain, even when it's having dramatic seizure activity. One of the things that seems to separate that from these sorts of biophysical models is volume. Seizures and spreading depression take place at very different levels of extracellular volume versus the space that it's in, that it has to expand. So everything I just told you is an emergent phenomena from the dynamics. If any of us had thought we could do this, we would have done this decades ago. But it's invisible to us. It's one of the problems of dealing with nonlinear dynamical systems. You really do get properties out. You can't predict from the components. And perhaps in retrospect, some of you could tell me that you could have done it with the additional conservation principles, for instance, that we put into this, but no one did. And this is really an accident to stumbling into it. I think a very useful accident. But think of what some of this implies or enables us to do. We have to now control not just a set point, for instance, but you've got to control through this landscape. There's a lot of regional interdigitation between what's normal, what's pathological. So you're controlling through a landscape. If I want to optimize energy use in a controller, this enables you to also do that, but I can now reconstruct the energetics of the brain. I can stimulate your brains enough to kill neurons. You do that naturally if you have severe epilepsy, and that we've shown in many different scenarios that serious enough epilepsy will burn out highly vulnerable neurons that need are kind of on the edge of where their energy supply is, and that have high energy needs. You can't measure that. I mean, you could in an optical experiment. We actually don't have the means to technically do that very well. But if you have a framework that enables you to calculate the energetics of the material you're working with, you can now begin to set safety limits, which have to be important in the way we interface with the brain. Think about what that means. If I have someone who's not having a seizure or a migraine, now that's most of the time for most people, even when they have serious illnesses, then what I want to try to do is avoid getting into such conditions. That also is a different version of control than what most of us are used to. Similarly, if I have someone with spreading depression and I want to avoid a migraine, what I don't want to do is let the controller drag them into a state of intractable epilepsy. So the nature of control, the control principles you'll use. Do I want to get to steady state in finite time as fast as possible but avoid any of the really pathological regions that are worse than having the migraine? Yes. All of this needs to be considered, perhaps, if we're going to make controllers for such conditions. In 1999, Vitsa Wadman was presenting this poster that would be published the next year at the Society for Neuroscience. There are some good things at the Society for Neuroscience, and a lot of it isn't in what people say. So they were taking these models of very large extended neurons, and what Kager, who was the student at the time, and Wadman and Samjian had done, is to try to do a canonical model of an extended neuron surrounded by glial cells that could mimic spreading depression. And they predicted, they didn't actually talk about it in this beautiful paper, but you'll see it in the diagrams that they have. There's a polarization axis that seems required in such model embodiments to generate spreading depression. You know, at my maturing stage, I don't have room for a lot of 15-year projects. But I've been trying to do this now ever since I left this man's poster. And we've had several grants on this. One of the problems of trying to do things when you don't know what you're doing or you don't have any guidelines before, it just takes a couple of false starts. So we had, actually with our colleagues in Germany, ended up with a German academic exchange project, which then led into a collaborative research and computational neuroscience project. We still didn't know how to get this right. But this is Ying Xiao's work. These are tangential slices of cortex. The reason I say that a few times on this talk is that they're relatively isotropic. If I come in from the side, I can do very different experiments. But this is about as isotropic a network as I can get out of any part of the brain. If you want to do a physics or wave experiment, or you're interested in symmetries, you need the ability to study that. And what Ying had shown in a chamber where we had a ring electrode deep and then a larger ring electrode up on top, so at least through the middle of it, I could put fairly uniform electrical fields going up through the cortex or down through the cortex, and all the neurons are pointing up and down. She showed that we could generate spreading depressions and begin to slow and perhaps break these waves up if we modulated them according to what we thought might be a way to control them. This is an unpublished but very new movie from Andrew Whalen. This is two different experiments. The top is control. We inject just a little bit of potassium chloride into cortex, and we're going to apply an electric field in a certain way. And we can now repetitively block these things with what I think is a very reliable set of statistics coming out. And all of this is related to the initial modeling. There's a caveat here. We haven't published this, and to protect Andrew and his upcoming paper, I'm going to just be a little cagey about what you'd all need to run back to your labs and duplicate this immediately. But this is pretty neat. This gives you the ability to control not just seizure activity, which we've worked on for a while. There's a paper I have in Journal of Neuroscience some years back now looking at proportional integral control. The integral part was important in setting up that controller for reasons which I'm happy to discuss. But you can develop empirical controllers that interfere with seizure dynamics. You can do fundamental controllers based on the kind of unification framework I've shown you. But this is the first time we've, I think, got the modeling right. I say that carefully because Grafstein in 1956 did some just amazing experiments on control of spreading depression. I mean, if you're working in 2017 and you read this line by line, it's so annoying. There are experiments in her paper that she just never illustrated and discussed. I don't know if she actually saw what I'm showing you now. She describes other experiments, but boy, it's the closest thing to a near miss from 70 years ago that I can think of. Anyway, I'm very intrigued that that state space control scenario that we're showing might be reliable in different regions of the state space to actually control different states that you would observe in a data assimilation framework and then manipulate into what would be non-pathological states. We've just spent about 6 months getting our N's nice and high so that our colleagues will think this is robust and reproducible, including us, and looking at the expected dose response curves that you would want to see that what I'm talking about actually is reasonably robust. So anyway, I hope within the next 6 to 12 months that this is all published. This is something that we also did. Most spreading depression and seizure experiments haven't been done in spontaneous awake animals. My colleague Bruce Gluckman has engineered a system that enabled us to record from mice. This is post-malarial epilepsy. This was just published a few months ago. But enables us to record for months on end without missing any bites. And it looks quite interesting. On the top is a seizure which lasts a good 30, 40 seconds or so in the animal and stops here. And following that on a slow time frame, so this is 10 seconds. Oh, I'm not interviewing that person now. This is 50 seconds. You see spreading depression slowly propagating from electrode to electrode. The seizure stopped a minute ago. These things are not that unusual, but it's very hard to get the electronics right to look at these very slow potentials. We throw most of this out. Here's another example. This animal actually dies. And there's a paper by Jeff Nobels in Science Translational Medicine a year or two ago offering the suggestion that in many different kinds of experiments that they did, that people die after seizures because spreading depression gets into the brainstem and the animal or the human stops breathing. So there are a lot of things that I think we can now do in looking at the interplay between seizures and spreading depression. My guess today is that they're intimately linked, and they just demonstrate the breadth of the repertoire that neurons and their networks can do under different conditions and different temporal trajectories. We've been hammering on micromagnetics to also try to, there's a reason for this beyond just that it's interesting. In terms of the actual physical interaction with patients who need to have implanted devices. My colleague Srinivas Tadikadapa is just amazingly creative at creating these tiny little MEM-scale sensors. This is a little diving board that has a magnetorestrictive material on it, and it, the end of it goes into the input of what is essentially a transistor here, and you can measure very small fields, but we're trying to do all of this at ambient temperature. You don't want to put cryogenic sensors or hot atomic magnetometry sensors into the brain. There are also diamond defect if you're a fan of all this stuff, but the nitrogen vacancy diamond materials have to be activated by the same microwaves that you use to cook food in your microwave. Probably not good for long-term use in Cortex. We're working with all of these at ambient temperature that can be actually put into the body. And then my favorite are what I call TWCs, teeny weeny coils. That's enough of a discussion of that for right now. This is a systems engineering workshop. Are brains observable? There's formal metrics to put on that. Are they controllable? As any of you who are in engineering here would immediately be asked if you were designing something new. Ching-Tai Lin, in his doctoral thesis at Maryland in the early 1970s, looked at the structure of networks that would be controllable using Kalman's basic linear approach. And he came up with this fundamental topology. I don't know if he had ever been to the American Southwest and seen one, but he called this a cactus, and the name has stuck. And it permeates a lot of our control engineering literature. In principle, a cactus, this is also a cactus, you can put control into a network and you know exactly what each of the nodes in the network will do. I can do a transform on that control of the actual adjacency matrix of the points and reverse it. A much more obscure paper will prove that these same topologies are observable from one point. You can fully reconstruct state. And if you go into the archaeology of the control literature, I love this. So Lin's paper is a classic. It's been cited over 500 times. It's in all the textbooks that deal with this. One of the problems from that is if you look at nonlinear networks with symmetries, it fails at some point. I mean, they're very careful to point out, Lin is very careful to point out that you can't have anything like dilations, meaning reflection symmetries in networks that you apply this to. And really, his strategy looks at the zeros where there are no connections as opposed to where there are connections. A little bit of philosophy on that. Rubin publishes this paper in the early 1970s. This is so hard to read. We ordered up his doctoral thesis from Columbia University to try to figure out what he was talking about, and I think we're the first people who ever checked it out. We blew a lot of dust off that thing. And in it are the group theoretic theorems dealing with electrical circuits at the time that help resolve some of the paradoxes that we were seeing in looking at neuronal networks. What do I know about group theory? Yeah. I talked to Jeff once, and Jeff says, oh, yeah, I know all about this. Thanks, Jeff. That doesn't help me. So I looked it up. There's this great set of books, and in this chapter by James Newman, he explains the theory of groups is a branch of math in which one does something to something and then compares the result with the result obtained from doing the same thing to something else or something else to the same thing. And I thought, oh, this is great. I understand it now. The same Andrew Whalen who brought you these amazing new control experiments ends up figuring this out. I don't know how in one brain he can do both of these things. This just was published in 2015 in PRX. The class meaning reflection or cyclic, of the permutation symmetry group determines, not just that it's symmetric or not, determines whether you can observe and control a network that is nonlinear and has symmetries. I mean, it just drops out if you read Rubin and could understand it. Cyclic symmetries are always observable and controllable. Lin knew that. Somewhere deep in that paper, it's obvious when you read it. He had the feeling that that was true, but he never said it, and he didn't have the formal basis to deal with it. Anyway, this is something else that we've recently come up with, but this applies to anything you want to work with. So I could take this very basic analysis of what is structurally controllable and observable and apply it to any feedback system. Now, if I look in your literature on controller design of networks, I don't find a lot out there. If any of you know what I'm missing, please let me know. But if I want to build here, for instance, something that I'm also working on, which is control of epidemic diseases, using a predictive control model and feeding in data from satellites, feeding in microbial analysis, and trying to then look at outcomes and feedback into my ensemble Kalman filter, you have to take the symmetries into account to make it controllable and observable. I don't see a lot that we've done as a community addressing that, and that's important whatever system it is that you might wish to work with. It also applies to policy, because that same feedback control is how I would want to design public health interventions if I'm not just optimizing treatment, but I'm trying to prevent some disease from occurring. Happy to discuss that in more detail. And I'll leave you with one last thought. A few years ago, we started reconstructing the childhood brain growth curves. It's a fascinating situation where most of your brain grows between birth and 2 years of age, and that was the period that there was almost no data. So there was literature talking about, well, from 5 years old on, the brain doesn't really statistically grow with the uncertainty that we have. You know, most kids start out small and they get big. And I know their brains are small when they're born. So we actually went, Jason Mandel did a whole series of these papers, all published as a group, reconstructing for both males and females. That's an old, wonderful problem in biology. At the turn of the 20th century, there was a major push by Goddard and others who were leaders in American psychology to prove that women's brains were smaller than men's brains. They are, but their body size accounts for that. And Stephen Jay Gould wrote a wonderful book, The Mismeasure of Man. I make my students read it. It's a wonderful accounting of that history and how our science really can cause a lot of damage when we're biased in all sorts of ways. And we're all biased in all sorts of ways. Anyway, what we're able to do and what we've been trying to do is to take these brain growth curves as trajectories. So you could take a given patient at some point with a small brain early in life and ask, what's the optimal trajectory to control to if I'm treating those individuals? So I actually do know something about hydrocephalus. Most of the stuff I've talked to you about today, you people in the audience know a lot more than I have ever learned or will ever know. But I know about these babies with a lot of fluid in their brains. And I'm just wrapping up now a randomized clinical trial that we've done with my colleagues Ab Kulkarni and Ben Worf. And almost all of these infants come in with small brains. And by taking these normal growth trajectories, you can see that their brains are growth arrested. And then with treatment, some fraction of them get catch-up growth, and some just don't grow at all. And if you look at the growth regulation literature, about 2005, Boris Schraman at Santa Barbara publishes a paper demonstrating how pressure changes can regulate growth in organs. Now, he's dealing here with Drosophila wings, but the point is all of your cells, as they divide, can vary in their rate of growth. If they grow a little too fast, you'll buckle the organ structure and feedback locally to slow down growth. And that's one way that most fruit flies have wings of equal size, antennas of relatively equal size, etc. I have wings of relatively equal size. This phenomena appears present in what we're looking at as pressure is relieved in these patients. And it is the patients who actually have catch-up growth and get into the normal range that cognitively do the best. This gives us the ability to manage people better, because if we're watching that brain growth, we can ask if they need to ride at a lower pressure. We do that all the time medically. We swap the valves out for lower pressure valves. And really, we focus on draining fluid in these infants. Our goal is to grow brains. So we're very excited about this. This is under second review, not quite published, but I thought I'd run it by you because it's so cool. Anyway, a lot of people, faculty and students and postdocs have contributed to the work that I've just shown you. I've got as many of the codes done archived. The links to these archives are all off of my website if you're interested, and if that helps you get a boost. And lastly, for the work with infectious disease, I've got a nice Washington University contingent that's also working with us on this, both from neurosurgery and in proteomics here. So thank you very much for your attention today.
I think we have time for some questions.
I didn't see any noise effects in your model. Is it important to include noise?
Oh, of course. And noise, I think from a philosophical standpoint, of course we have measurement noise. Right? And
Model noise.
what?
Model noise. When you look at the model.
Yes. So, but I'll start from measurement because it's easier. So we have real measurement noise. If you look at nervous systems, there are multiple sources of true randomness in the dynamics themselves. These membranes are thermal membranes. You can push them into states where, near threshold, where we think noise effects will actually determine whether or not it fires. That's an old experiment by Hodgkin and Huxley. Release of transmitter. There's not a lot of transmitter released at most central synapses, often one packet from one synapse. It's probabilistic. And so there's true noise in the system at central synaptic transmission where your decisions are being made. It's very different from a muscle. I decide I want to move my hand. I get a big flood of transmitter onto those muscles. There is not a lot of question that I worry that my muscle movements are probabilistic, but my central processing is. And so there are true central noise components. The last issue, though, is model uncertainty, and that's a ubiquitous problem for everything I showed you today. There are some really interesting ways, I think, of handling that. There's a paper by Bacon colleagues, again from the Maryland group, Ed Ott and others. I actually put a whole chapter in my textbook on this because I think it's central, and I don't think any of us have properly put this into the neuronal version of quantifying uncertainty. But one of the things I like doing, and I have my students do in classes, is to actually parameterize model uncertainty. Some of the very basic issues of how do you embed the dynamics. Do you want to assume that there's a geometric transformation that you got wrong? Do you want to assume that there's drift you're not accounting for? Those issues also pertain to whether you're doing machine learning. And so handling what is unobserved and unaccounted for is much more involved, I think, than simply ascribing it to noise. I think that's sort of the essence of it you're also asking me. I think if there's any one frontier to work on right now, it might actually be trying to handle uncertainty in these simple models when you want to interface them with very complex structures like brains.
Take one more.
So you talked about the volume expansion that occurs when some of these ion transports occur, and then later on you talk about much later, mechanical feedback and development. Is there mechanical coupling between neural circuits? I mean, it seems like there could be. If they have to share a volume, only one can trigger it at a time.
If you're asking if the volume expansion, which goes on all the time, might give mechanical coupling, that's a great question. I haven't thought of that. But I hope I can steal that question and think about it some more over time. There are mechanical effects of deforming neurons and membranes. There are certainly mechanical effects on growing and developing cells that have been very well studied. But I don't know of anyone who's looked at the direct mechanical of your neurons as you're sitting here. Most of you at least are just constantly expanding and contracting as they get more or less active. I hadn't thought of that. That's a really great question. I have no idea what the answer is.